Well, I think we're going to make a start. We've got loads of people already starting to come in. So thank you guys so much for coming to this is week 2 of our free online biology course. And thank you for taking the time out of your day to join me here to help you learn A-level biology. I've really been blown away by the response, to be honest. So many people have messaged me, asked me questions, and I can only thank you guys so much for responding well to it and sending me feedback. The feedback's been amazing so far, so really thank you very much with that. So last time we were doing photosynthesis. So just for anyone that's new here, my name's Ephraim. I'm a medical student studying at Barts in London, and I've been a private tutor for about 3 years now, but since lockdown started, I know so many of you are struggling to keep up with your workload, especially with biology. It can be difficult not learning when you don't have your teachers around, you don't have any kind of structure. So that's why I'm here to help you with that. And we're going to be doing free weekly biology lessons to try and get you up to speed with what you need to know. So I've been teaching for over 300 hours plus now, and I've got a lot of experience, and I know what works for students. I know the kinds of things that I do come up in the exam, and I'm going to try and share my secrets with you. So as many of you know, UCAS is open now. So if you guys have any questions to do with the medical school application, please do get in touch. A-levels are hard, as I said last week, they are very hard. But if you guys have some kind of motivation to keep going, that's the best way to stay motivated through this really tough subject. But most of all, if you enjoy the subjects, everything becomes a lot more easier. If you start to really gel with the content, start to understand the content, you will, trust me, you will find the content a lot easier, and you will start to learn everything a bit better. So I've done some exam and revision tips last time, so I won't really bore you with that. But if you haven't actually seen it, go on my YouTube channel. Our week 1 recording was all live. So photosynthesis, let's get started. So as a quick recap, what did we start looking at? We looked at why biological processes need energy in the first place. We looked at different examples. For example, spindle fiber formation is a good example of why loads of biological processes need energy. We looked at this really key molecule called ADP, adenosine triphosphate. We looked at some of the key reactions that are involved. We looked at that main one, which was called photophosphorylation. So phosphorylation is basically adding a phosphate group on. Photophosphorylation is just doing the same process, but using photons or light energy. And if you need to recap any of those reactions, they're all on YouTube. So really key concept, plants, we call them autotrophs. Autotrophs basically mean they automatically make their own food or glucose by photosynthesis. And this, by the most part, happens during the daytime due to those light-dependent phases that we'll talk about shortly. Mammals, on the other hand, they're heterotrophs. They rely on autotrophs. So we basically eat autotrophs and get the glucose from them and start to respire them. And overall, we saw that equation. Again, you can look at that online. So our next topic will probably be respiration, because it's a good way to end photosynthesis, because photosynthesis, respiration, they're basically opposites of each other. One's making glucose, the other one's using that glucose to make ATP. Okay, basic concepts. These are some really key terms that we looked at last time. And one thing that we'll be talking about a lot today are these redox reactions. Okay, so the best way, everyone's heard the acronym or the mnemonic from GCSE, that oil rig method to remember everything. So we're just going to quickly recap that. So oxidation reduction. You can talk about oxidation reduction in many different ways. Obviously, the most obvious one is in terms of oxygen. As the name suggests, oxidation, or oxy, means the adding of oxygen. And reduction, obviously the opposite, is removing oxygen. That mnemonic says oxidation is the loss. That basically means the loss of what? Electrons. So when I say oxidation, I'm in terms of electrons, I'm basically saying it's the loss of electrons, or the reduction is the gain in electrons. The last one, probably the most important one, is in terms of hydrogen atoms. Okay, so the best way to remember it is electrons and hydrogens, they're the same pattern. So if oxidation is a decrease in electrons, hydrogen atoms are also a decrease in hydrogen atoms. Reduction is therefore a gain. We're going to talk about loads of coenzymes, which are these molecules that basically aid in the transfer of different molecules from one area to another area. So we were introduced to this NADP molecule. We saw how it transported hydrogen atoms from the light dependent stage. And now we're going to look at the Calvin cycle, where those hydrogen atoms are taken to. So if I say the reduction of NADP, all I mean by that is adding a hydrogen atom to NADP. And so you guys don't get confused with NAD, which is what we're going to see in respiration. The way to remember the coenzyme used in respiration is NAD. The coenzyme used in photosynthesis is NADP. And the way to remember it is P, that's just photosynthesis. Okay, that's the best way to remember which coenzymes are involved in what reactions. Okay, so as always, if you guys have any questions, any messages you want to send them on the chat, send them on the Q&A section. I can try and get to them as soon as possible. What is your YouTube channel called? If you go onto my Instagram, there's a link to Google Drive. All the instructions will be there and you'll be able to find it. So this session, yeah, is mainly focused on photosynthesis, our first topic. We'll then move on to, I think, immunology or respiration next. Okay, so we looked at the chloroplasts, the structure of it. Again, look through the YouTube channel, you'll find everything. But the most important things I want to highlight are these thylakoids. That's what we're going to look at a lot today, and especially the membrane of the thylakoids. Okay, so all eukaryotic cells have membrane-bound organelles, and the plant cell, being a eukaryotic cell, has a membrane-bound organelle. And even inside this membrane-bound organelle of the chloroplast, there are even more further membranes. That's just a way to classify different organisms. Let me just... Okay, so we looked at the different parts. I also gave a quick explanation on why chloroplasts actually have their own circular DNA and starch grains, and I talked about the phylogenetic parts of the chloroplast. So again, look at that. So we also looked at the wavelengths of light, the absorption spectrums, action spectrums. Photosystems. Photosystems are really important to understand. So I'm going to bring up a quick revision guide I made, which should help you to understand everything. So this revision diagram is available for free on the Google Drive. Just a couple of things I want to highlight. Let's look at the photosystem. Photosystems that are always found on the thylakoid membrane. So this black line represents our thylakoid membrane. We have that photosystem 2, photosystem 1, both work at different nanometers of light. So we have photosystem 2 works best at 680 nanometers of light. Photosystem 1 works best at 700 nanometers of light. In between photosystem 2 and photosystem 1, we have an electron transport chain. Very important thing to understand. Basically, what's happening is, let's quickly go through it. We have an electron that's raised to a higher energy level by the photons from chloroplasts, and especially the primary pigment reaction center. That high energy electron is just basically moving through our electron transport chain proteins, and the energy lost in those processes is used to actively transport protons from the stroma into the thylakoid space. Then when we've got high concentration of protons, they move back down through ATP synthase to generate ATP using that proton motive force. If any of that doesn't make sense, everything will be online. These diagrams will also be online. Let's get back to the PowerPoint. Okay. So last time we looked at the stages of photosynthesis. I'm going to quickly go through that because I know some of you are new here. So we have our light-dependent stage. Uses light, obviously. So we're going to represent these arrows as light. This is just a brief roadmap of all the reactions involved in photosynthesis. There's two main reactions. We have our non-cyclic photophosphorylation and our cyclic photophosphorylation. So non-cyclic and cyclic photophosphorylation. They both work in quite similar mechanisms, but they produce different products, okay? And we're gonna look at what those products are. They basically make different products, for example, reduced NADP, ATP, which then go and feed into our light-independent stage, which is our Calvin cycle. Our Calvin cycle is what makes glucose, okay? So the electron transport chain, we've already looked at non-cyclic photophosphorylation. Just for completeness sake, I'm going to quickly draw it. So remember, these diagrams I'm showing you, they are visual prompts to try and help you to understand everything that you need to know from these. In a textbook I've seen, it's about 30 or 40 pages just describing these different topics. And it's not worth remembering all of that knowledge. I've looked through all your specifications. I know exactly what's going to come up, what they're going to ask you. And these are the key things. I'm going to summarize them in a diagram. So these diagrams, basically, I want you to draw them out, make some notes on the side, and daily, if you can, try and draw out these diagrams. If you draw out these diagrams daily, you're going to remember them for a long time. Now, I made these diagrams about 3 years ago, and I can still remember them. The whole of photosynthesis, respiration, I've learned them all, okay? So let's just quickly start. Photosystem 2, represented by that box. Light enters photosystem 2, so we're going to represent them as arrows. It then travels to the light harvesting system, which is your chlorophyll B, your carotenoids. It then passes into our chlorophyll A, or our primary pigment reaction center. Okay? Once it's in there, it's going to excite those electrons using the photons of light. So I'm going to represent the up arrow as the excitation of electrons to a higher energy level. They're going to move down that electron transport chain. Okay, that's that down arrow. The electron's losing energy, hence is a down arrow. We're then going to make ATP from this process. That electron is directly going to feed in and replenish photosystem 1. Okay, and more light is going to enter, re-excite that electron found in chlorophyll A, or that primary pigment reaction center, to an even higher energy level than before. We're going to add it to a proton and NADP to produce NADPH, or reduced NADP. Never say reduced NADPH, doesn't make sense, just cancels each other out. It's either reduced NADP or NADPH. This process, that electron that we've looked at over here, this electron is eventually, all the electrons in chlorophyll A, especially in photosystem 2, are going to run out. So we need to replenish the source. The way we're going to replenish those electron source is by breaking down water into 2 protons, 2 electrons, and half an oxygen molecule. That's the photolysis of water. Photolysis is basically using photons to break down something. Lysis just means breaking something down. Photolysis is breaking something down. And in this case, it's going to be water. Those electrons are used to replenish chlorophyll A, protons using the electron transport chain, and oxygen, that's for us. That's what we can breathe. And you'll see that in the overall reaction of photosynthesis. That's a quick recap. If anyone has any problems with that, let me know. The products, this is where it gets a bit important. The products of non-cyclic photophosphorylation, one of those reactions involving photosystem 1 and 2 in the light-dependent stage, produces ATP. ATP is using the Calvin cycle, reduced NADP is using the Calvin cycle, and the photolysis of water we discussed it. Okay. The next reaction, this is where we need to pick up from last time. So in the light-dependent stage, which uses lights, there's 2 reactions. So non-cyclic photophosphorylation, your cyclic photophosphorylation. This word cyclic is really important. You'll understand why this whole thing is cyclic. If you look in your book, everything just looks like diagrams. Nothing makes sense. They don't actually tell you why it's called cyclic. I'm going to tell you why. These diagrams, again, this is a prompt diagram to help you to remember everything. So draw along with me and you'll understand where I'm coming from. So cyclic photophosphorylation only uses photosystem 1. That's really important. So non-cyclic photophosphorylation uses both photosystem 2 and 1. Cyclic only uses photosystem 1. It's really important. P700 nanometers. Light enters photosystem 1, passes through the light harvesting system into your primary pigment reaction center, where that electron is then excited to a higher energy level. Just like this, it's the same start as non-cyclic, except we're using photosystem 1 only. That electron is then going to move down the electron transport chain, just like before. This time, it's going to also make ATP. Again, just like the previous non-cyclic stage, we make ATP. That's why it's called the electron transport chain. And if you're not too sure about any of these electron transport chain things I'm talking about, please go back to the previous slide or look at the YouTube channel, you'll find an explanation. But yeah, it makes ATP. This is where it changes. Normally in non-cyclic, you would see over here your photosystem 1 or the other photosystem. This is where it's different. This electron then goes and feeds straight back into chlorophyll A of photosystem 1, where it first originated from. I know that looks a bit like a triangle, but this is where that whole cyclic thing comes from. The electron is moving in a cyclical fashion, straight out of chlorophyll A. It gets excited, goes down the electron transport chain, and then exits again back into chlorophyll A. And the reason it does that is because cyclic photophosphorylation only relies on photosystem 1 and only makes ATP. That's really important to understand. Only makes ATP. In the previous step, non-cyclic photophosphorylation, we saw the production of reduced NADP and ATP. This is not the case. We only make ATP in cyclic photophosphorylation. And that's it. That's the prompt diagram that should help you to, in the exam, understand this. So what I used to do in my exams, I was quite lucky. In my exams, I had a whole 3 pages just on these reactions and asking about the different products. What I did, I quickly sketched out all these diagrams. It took me about 1 minute at the back of the paper and the extra space, quickly sketched out all the diagrams. And then when they asked me, for example, what photosystem does cyclic photophosphorylation use? That's in my diagram. It uses photosystem 1. then they asked me, what are the two products from non-cyclic photophosphorylation? Drew out those diagrams. I knew it was reduced NADP and ATP. That's why these diagrams are so helpful. You don't actually have to tire away reading away all those pages in the book. You just have to remember these diagrams, and these act as a prompt. So let's go through it one more time. Try and draw it along with me, flip the page over, try and see how much you remember. So we start with our box with a 1 inside it, representing photosystem 1. Photons, or light energy, enters photosystem 1, passes through the light harvesting system, through that into the primary pigment reaction center where chlorophyll A is. Chlorophyll A contains electrons. Those electrons are then excited using the light energy to a higher energy level. And again, all the notes are on the next slide. That electron then goes down, losing its energy down the electron transport chain to produce ATP. Okay, that electron has now returned back to its original state inside the thylakoid space, and it's lost all of its energy in the electron transport chain. That electron is now going to go and feed back into chlorophyll A to replenish the source of electrons. This way, cyclic photophosphorylation of photosystem 1 does not need any photolysis of water. There's no photolysis of water. And the reason is because we had photolysis in the previous stage to actually replenish the source of electrons. There's no need for that in this step because the electron is constantly going in a cyclical fashion. There's no need for photolysis. That diagram's it. That's all of cyclic photophosphorylation. Does anyone have any questions before we move on to the next step? Let's have a look at is this content for OCR A2? It's designed for all the exam boards, okay? Up to this point, it's exactly the same for all exam boards. Any changes, I will let you know in the slideshow. But yeah, this is for all exam boards. Let's have a look at it. Okay. Yeah, I haven't got any messages, any questions, so I'm assuming everyone's happy with that. If you have any questions, let me know, we can go over it. That's cyclic photophosphorylation. The notes are on the previous slide, which I'll put on the Google Drive. These are the products of photophosphorylation. Okay, products are ATP, only ATP, no reduced NADP, only ATP. ATP is used in the Calvin cycle, which we're going to look at next. The electrons, they're not passed on to NADP because that electron goes and feeds back into chlorophyll A, acting in that cyclical fashion, hence cyclic photophosphorylation, and replenishes the source of electrons. Okay. Let's move on to the next step. Okay, this is the Calvin cycle, probably the hardest bit. So just before I start, is anyone on the Edexcel exam board? If you are, can you please raise your hand or just make yourself known, because it slightly differs. Edexcel slightly differs from AQA and OCR. So we've got one person at Edexcel. Okay, that's fine. We'll go through it. A couple of people on Edexcel. So yeah, we'll go through both steps. They're pretty much the same thing, just a couple small changes in the wording. Okay. But the Calvin cycle is another way to say the light independent stage. So we've talked about the light dependent stage, which relies on light or photons. The light independent stage doesn't rely on light, but it does rely on reduced NADP and ATP, which come from the light dependent stage. So technically, it also does rely on light. Key points. This whole Calvin cycle takes place in the stroma of the chloroplast. So this is the fluid surrounding those thylakoids, those granum. And what we're going to make from this, the products of this, is basically glucose. But there's intermediates are really important. They differ between the three exam boards. If you're on AQA and OCR, just remember triose phosphate and ignore GALP. Okay. If you're on Edexcel, remember GALP, forget triose phosphate, and you'll see why in a minute. Okay. But if you're on Edexcel, remember GALP, ignore triose phosphate. If you're on OCR Edexcel, remember triose phosphate, ignore GALP. Okay. So what do we need? What are the reactants for a Calvin cycle? There's four main things. Reduced NADP, we know where that comes from, the light dependent stage. CO2, CO2 comes from us. We exhale CO2 as a product of our cellular respiration. That then enters through the stomata, those guard cells open up, the CO2 gets into the leaves, and that's used as one of the reactants in the Calvin cycle. We have a new molecule called RUBP, ribulose bisphosphate, really important molecule. Ribulose bisphosphate. Ribulose bisphosphate that's the full form of saying it. If you want to say it in its abbreviated form, say R capital, small u, capital B, capital P, RUBP, ribulose bisphosphate. Okay, and ribulose bisphosphate is found inside the stroma in high quantities of the chloroplast. Okay, it's found in the stroma. It also needs ATP. ATP comes from the light-dependent stage. So this is probably what you've seen in any good textbook. It looks really complicated. There's really big names here, and we can't really memorize that. But it's a cycle. That's what I want you to take away from this. Let's do a similar diagram. These are prompt diagrams to help you to remember all the steps in the Calvin cycle. So again, it's a prompt diagram. This holds no value in the exam if you write it as an answer, but it helps you to remember all the steps using these diagrams. So I know a couple of you are on Edexcel, so just bear with me. We're going to do the AQA and OCR version first. The majority of it's the same, and I'll tell you until where you can stop listening, and then, yeah, just bear with me. So the Calvin cycle. The Calvin cycle, as the name suggests, is a cycle. It's going in a cyclical fashion to produce glucose, okay? So let's draw this diagram. The first step of this diagram is carbon dioxide. That's from what we exhale. That carbon dioxide is going to join with something called ribulose bisphosphate. Okay, so I'm going to represent that with an R. So R, ribulose bisphosphate, C is your CO2. It's really important to understand how many carbons are involved here. Ribulose bisphosphate is a 5-carbon molecule. So I'm going to put 5C. Carbon dioxide is a 1-carbon molecule. So first step, ribulose bisphosphate, RUBP, which is a 5-carbon molecule, combines with CO2, which is a 1-carbon molecule. We need to basically combine them together to make a 6-carbon compound. So 5 carbons, 1 carbon, 6 carbons. So we're going to make a 6-carbon compound from this. So I'm going to represent that with a big 6 around it. That should help you remember it's a 6-carbon compound. You don't need to know the name of the 6-carbon compound, just say in your exam 6-carbon compound. So to join ribulose bisphosphate and carbon dioxide, we need an enzyme. This enzyme is called rubisco. And again, you need to write it in a special way. Capital R, capital B, capital C. You capitalize the first word. That's rubisco. It's an enzyme that catalyzes the formation or the combination of ribulose bisphosphate and carbon dioxide. The full form of rubisco, you don't really need to know, but if you're interested, it's called ribulose bisphosphate carboxylase. Yeah, it looks like a really daunting word. Basically, ribulose bisphosphate is your RUBP. We're going to add a carbon to that. That's where you get your carboxy from. And anything ending with an ASE is an enzyme. That's where we get our carboxylates from. So ribulose bisphosphates plus carbon gives you a 6-carbon intermediate. Just say it's a 6-carbon compound. That's what we need to know. That 6-carbon compound is very volatile. Can't really stay as a 6-carbon compound for too long. So what it does, it splits. It splits into 2 3-carbon molecules. So we've got a 6-carbon molecule. We're going to split into 2, exactly the same 2 3-carbon compounds. And they are called GP, or glycerate phosphates. I'm going to represent that with 2G. So G equals glycerate phosphate, or GP. And it's perfectly acceptable to put GP in your exams. But if you just want to be sure, put glycerate phosphate as well. So that 6-carbon compound is split into 2G, or 2 GP molecules. And that's a 3-carbon compound. GP doesn't really resemble glucose, which is our end product, very well. So what we're going to do is transform G into something else, our GPs into something else. That's where it gets a bit different. So if you're on AQA and OCR, or for all the exam boards, up to this point is exactly the same. If you're on Edexcel, it changes slightly. Okay, so our 2 GP molecules, they're going to be converted into 2 different molecules that resemble glucose a bit better, and they're called TP. So I'm going to put 2T. T equals TP, or triose phosphate. Triose phosphate resembles glucose a bit better. So we're going to basically convert GP into a different molecule called TP. And then we're going to represent that with 2T. So 2 TP molecules. Again, TP, or triose phosphate, is a 3-carbon compound. So just be aware the distribution of carbons in this whole Calvin cycle. But to transform GP, or to convert GP into TP, it needs some things. It doesn't happen spontaneously. We need to give it some things. The first thing we need to give it is 2 hydrogen atoms. Where are we going to get those hydrogen atoms from? reduced NADP. We've been saving up, we've been collecting all that reduced NADP, we're going to spend it all here. So for the conversion of each GP into TP, we're going to need one reduced NADP molecule or one hydrogen atom. So for 2, we need 2. And I'm going to represent that with a double-headed arrow going in to the conversion arrow. That basically is representing NADPH. And we're going to give those 2 hydrogen atoms from reduced NADP, we're going to oxidize it, give its 2 hydrogen atoms to GP to help it to convert it to TP. That's all we're doing. And what we get out of that is NADP. NADP, we're going to be making NADP from this process by removing the hydrogens, we're oxidizing it. That NADP can go back into the thylakoid space and work back into the light dependent stage to collect more hydrogen atoms for future Calvin cycles. That's where the NADP goes. To convert it, we also need some energy. Energy we're going to get in the form of ATP molecules. So I'm going to represent 2 ATP molecules being input into this process to provide energy by these 2 dots. Remember these dots. We're going to see loads of dots that represent phosphates or ATP in my future diagrams regarding respiration. So it all does make sense. The arrows represent the input of mainly hydrogen atoms. These arrows with a dot on the end represents the inputting of ATP. So we've converted GP into TP successfully now. And that has used 2 reduced NADPs and 2 ATP molecules. That TP can now combine together to form glucose. So TP is a 3-carbon molecule. Glucose is a 6-carbon molecule. So all I'm going to do is combine 2 TP molecules, stick them together to make a 6-carbon glucose. That's where your glucose comes from. TP can also go on to make things like fatty acids. It can also make amino acids, loads of other things that are needed for the plant to grow and survive. Okay, so TP, a really important molecule. It basically makes your glucose, your fatty acids, and your amino acids. In certain textbooks, I've seen that they also say GP helps in the formation of fatty acids. That's also true, but I need to make a compromise between all the examples. But the main thing you should understand is that TP makes glucose, and if you're an OCR, GP makes fatty acids. If you're on other examples, TP makes fatty acids. Hope that makes sense. So we've now made our glucose by combining 2 TP molecules. That's it, we're done. But this is a cycle. If I don't replenish those reactants that we started with, that RUBP and CO2, this cycle can't carry on. So we're going to regenerate some of these things. So we're going to use some of the TP that we've just made, and we're going to regenerate RUBP. So if we stick on some TP molecules together, we can actually make RUBP. And for that process, we require another ACP molecule. Okay, so the conversion of TP to regenerate, this is really important, it's the regeneration. It's a really important word to mention in your exams. Regeneration of RUBP requires some TP molecules, don't worry about how many, but it requires TP molecules. And it also requires an ATP molecule. We've made RUBP again. That can go and redo the cycle again. That CO2 just enters the stomata one more time and is used in the Calvin cycle. And that's it. That's the Calvin cycle. And that's all the reactions in photosynthesis done. So let's recap that one more time. So again, these are prompt diagrams. The more you write them out, the more they'll stick in your memory. I explained the science behind using diagrams and pictures to help you remember in my previous webinar. So it uses a different half of the brain that's not really well utilized because we rely on remembering things from words, which is already pretty much fully occupied. So we're going to use pictures, exploit our memory of pictures to help us understand these really complicated reactions. One more time, the Calvin cycle. RUBP plus CO2. RUBP is 5 carbon, CO2 is 1 carbon. We're going to join them together to make a 6-carbon compound, so you put a big 6 around it. This process uses ribisco, which is an enzyme. Ribulose bisphosphate carboxylase. Ribisco is catalyzing the formation of that 6-carbon compound from your RUBP and your CO2. It's volatile, that 6-carbon compound. We're going to split into 2 GP molecules. GP is then converted into 2 TP molecules. And the conversion of your 2 GP molecules into your 2 TP molecules requires, those double-headed arrow represents 2 reduced NADPs, or especially the hydrogen atoms. So we're going to oxidize, really important, we're going to oxidize NADPH to remove its hydrogen, and then we're going to add it onto GP to help it to convert itself to TP. This process also requires 2 ATP molecules. So we're going to put that by an arrow with a ball at the end to represent the input of 2 ATPs to help the conversion of GP into TP. TP goes on to make glucose. If you're on OCR, GP goes on to make fatty acids. But the main one for all exam boards is glucose. If you're not on, obviously not Excel, because it's just for AQA and OCR at the time being. Put that on top, AQA, OCR. But yeah, main one is TP makes glucose. TP is then used to regenerate RUBP. That process requires ATP. That's it. The only thing I need to mention, if you're in Edexcel, the main difference is instead of TP, you have a different compound. It's called GALP. GALP stands for, so long name, get ready for it, glyceraldehyde 3-phosphate. It's a mess to spell. Glyceraldehyde 3-phosphate. You don't really have to remember that. Just remember GALP. But yeah, glyceraldehyde 3-phosphate is used instead of TP, but it works in the same way. TP is used to make glucose and then used to regenerate RUBP in the same way that TP, which is found in your AQA and OCR, is the same thing. So if you're on Edexcel, one more time, you're using GALP, which is again a same 3-carbon compound instead of TP. If you're on AQA and OCR, you use TP instead of GALP. Just a few differences that you don't really know why, but they're just some differences in examples. But that's it. That's your Calvin cycle. Does anyone want me to go through that one more time, or should we move on? We're pretty much at the end already. Does anyone want me to go through those diagrams one more time? Any questions from those diagrams? All these diagrams, again, will be recorded. This whole thing is recorded, this whole webinar. The revision guide I showed at the beginning, as well as loads of other resources, are found on Google Drive. So if you're struggling, you're going to find it there. So anyone else, last call, do we need to go over Calvin cycle again? Good. No, let's move on to the next thing. So the products of the Calvin cycle. We made one glucose. That's it. We've made it. We've made our glucose molecule. We've also made other things needed for that plant, such as fatty acids, amino acids. But the main one we made is glucose. And that's the one you should be remembering. But in general, TP is used to make your glucose, your amino acids, things like that. Your GP, if you're on AQA and OCR, used to make your fatty acids. If you're on Edexcel, GALP is used to make all of that. Glucose, fatty acids, amino acids, all of it. So this will be a really satisfying end to respiration for you. Okay, what's going to happen is I'm going to put that equation that you've all mastered by now, that equation of photosynthesis, you've all learned that in GCSE. Now you're going to understand where everything comes from. So CO2 add H2O gives us C6H12O6 plus oxygen. To balance it, you remember you just add a 6 in front of everything besides glucose. Let's talk about where everything comes from. CO2 is a reactant. Where do we use CO2? We used it in the Calvin cycle to combine it with ribulose bisphosphate. Let's write that down. So we've used CO2 in the Calvin cycle. Where did we use H2O? We used it in the photolysis of water in the light dependent stage to replenish the source of electrons in the primary pigment reaction center of chlorophyll A. C6H12O6 is our glucose. You made it in the Calvin cycle. And oxygen, the most satisfying thing to understand where it comes from. When we did photolysis of water, which I'll write down here, is H2O goes to 2H plus, 2E minus, and half O2. For this step, we need light or photons of light. O2, that's where it comes from. The photolysis of water in the light dependent stage gives us the oxygen as a product of photosynthesis, and it's useful for us in respiration. Also, this plant can use the same oxygen it makes in photosynthesis for respiration during the nighttime. That's where you get your oxygen from. So from a GCSE student to an A-level student, you finally understood where all these little things come from the reaction. So well done to you, you've kept along. We've pretty much covered all the main content. Let's just do a couple extra practical things that you should understand some topics that the exam boards like to ask, because I think you guys should all be aware that the exam board loves to ask application style questions or suggest style questions. Everyone knows the facts involved in photosynthesis. They expect everyone to get the facts right. That's why they don't ask a lot of it. They ask a lot of application questions. So we're going to quickly go over some kind of things relating to that. So limiting factors. You've probably seen this concept of limiting factors when we talked about the enzyme, or if you did the enzyme topic in year 12. Limiting factors basically slow down the efficiency of an enzyme and stop it working at its maximum rate. So for example, the limiting factor of an enzyme could be its temperature. Then if the temperature is too high, it denatures the enzyme. That's a limiting factor. If it's an enzyme breaking down lipids into fatty acids, if the concentration of lipids starts to fall, that becomes a limiting factor. Same thing applies here. Limiting factors in photosynthesis can be things like light intensity. If there's a stronger intensity of light, you're going to increase the rate of photosynthesis, especially in that light-dependent stage reactions. And we're going to increase the rate. We're going to make more reduced NADP, more ATP, and eventually we're going to make more glucose. But if we reduce it, that can become a limiting factor. Wavelengths of light. We talked about that last week when we talked about the visible light spectrum. I said that the reason why plants are green is because they absorb all other parts of the visible light spectrum except green light, because plants don't have chlorophyll specialized to absorb green light. We have carotenoids, carotenoids. They are good for absorbing orange light. Your chlorophyll B is good at purple, blue light. Chlorophyll A, again, for yellow light, but there's no chlorophyll or no photosynthetic pigment that can absorb green light, so it's reflected. So if we're, for example, talking about greenhouses and greenhouse farming, you probably know that in the UK, currently it's the season for strawberries, okay? In the wintertime, it's not the season for strawberries. If we left them outside in the environment, strawberries wouldn't grow. The reason you get strawberries, mainly because some of them are imported, but we can also make them in greenhouses. And farmers or greenhouse farmers exploit the limiting factors of photosynthesis to increase or enhance the growth of these plants. So for example, they'd put really strong lights, a really strong intensity of light. They'll also change the wavelengths of light, so there's more purple, blue, yellow, and they'll remove green light, so that's increasing efficiency again. They, for example, put some lamps or pumps to pump in CO2. So that means we get more CO2 for the Calvin cycle. Temperature, they'd start increasing ambient temperature to a good enough temperature for the specific plants that they're growing. And that would benefit the enzymes and make them work at a higher rate. Two enzymes we've looked at in this topic are Rubisco. Rubisco is using the Calvin cycle. If we increase the temperature, we increase the rate of reaction for the combination of ribulose bisphosphate and CO2 using Rubisco. The other enzyme / channel protein we looked at was ATP synthase. If you denature this channel, electron transport chain proteins and ATP synthase, you're no longer going to make your glucose, your ATP. So temperature, again, a limiting factor. Stomata, I thought it's worth just bringing this up quickly. You'll learn about it in a different topic, but stomata are found on the underside of leaves, and they are opened and closed by these guard cells on the side, these guard cells. And depending on different conditions, they can either open or close. So CO2 gases, for example, enter through and exit through the stomata. So we need to make sure that those stomata open at the right time. Also, water vapor can exit through the stomata, so be aware of that. These are just some graphs to help you to understand the different limiting factors. So this graph I've taken from a book. It basically illustrates how light can become the limiting factor. From A to B, it increases, which also corresponds to an increase in the rate of reaction. It starts to plateau over here when at B. At B, the light intensity has become, is at its maximum, okay? So the plant's got enough light, but something else has become the limiting factor. So we've increased the light intensity to its maximum, but things like CO2, temperature, things like that have not reached their maximum. So that they become the limiting factor. It's important to remember at the plateau, so at B or at the plateau, something else besides this current factor becomes the limiting factor. Hence we see that plateau. That's light intensity. I'm not going to dwell on it too much because it's all in the notes here. Wavelength of light. So we talked about those action spectrums, there's absorption spectrums. So if we give the correct lights, wavelengths of light, so we looked at carotenoids, chlorophyll A, chlorophyll B, we looked at the action spectrum. If we give certain wavelengths of light, we're going to increase the rate of reaction until that becomes a limiting factor, and then something else, and until that becomes its maximum, then something else, for example, CO2, light, intensity, temperature becomes a limiting factor. Concentration of CO2, obviously if you raise the CO2 concentration, the stomata open, allowing more CO2 for the Calvin cycle. But again, if we increase it too much, the stomata will close, and that will prevent the expelling of oxygen and water vapor. So you need to kind of compromise that when we're looking at, for example, greenhouse environments or greenhouse farms and how they control their environments. You need to make a compromise between allowing too much CO2 to leave or too much CO2 to enter. So just be aware that it's a compromised condition. But in general, if you raise the CO2 concentration, you're going to increase the rate of photosynthesis up to a point, which is this plateau point, and then over here, something else becomes the limiting factor. CO2 is obviously used in the Calvin cycle. Temperature, we've already talked about it. Temperature can affect things like enzymes, so rubisco, ATP synthase, all those membrane proteins. For example, there's electron transport chain proteins, your phospholipid bilayers around your chlorophyll, your thylakoid membrane, all those things can become damaged, increase their permeability and reduce the rate of photosynthesis. So temperature, again, another limiting factor. And then one that you probably don't really think about too much, or that's not mentioned a lot in the textbook, is water stress, or conditions where water is not in plentiful supply. So the stomata of the leaf basically close when there's a shortage of water. Okay, that's an important concept, because normally water exits through the transpiration stream and leaves through the stomata, which is another topic in AS. But if you close these stomata, you're going to not allow CO2 to enter. So if this plant under water stress, stomata will close, CO2 can't enter, and then you won't get enough CO2 for the Calvin cycle. So again, a compromised condition. And it all revolves around the opening and closing of these stomata. So I've mentioned loads of different limiting factors. I also mentioned how it's a good scenario to talk about it, an application style question in an exam. And I've seen quite a few of these kind of questions where they say, what kind of factors would greenhouse farmers need to consider when trying to grow plants? And you talk about things like light intensity, wavelengths of light, temperature, CO2 concentration. You'd also talk about limiting factors and why especially things like CO2 concentration is so important. You'd mention the Calvin cycle. Temperature, that you'd mention rubisco, you'd mention the electron transport chain, you'd mention ATP synthase. Those are the kind of things you should be thinking about. This thing I've only really seen on OCR, but I think it's worth knowing for kind of application style questions for other examples. This slide is showing you how limiting factors we've talked about previously affect GP concentrations, RUBP concentrations, GALP or TP concentrations. So if you remember on GALP, it's for your Edexcel. If you're on AQA or OCR, it's your TP. So how these limiting factors can affect concentrations of these three key molecules. And so this is where these prompts diagrams really come into its own. If you draw out these diagrams, you can actually see what happens if you limit a factor. So let's quickly go over one example. For example, temperature. If the temperature is too low, we talked about enzymes start to denature, and electron transport chain proteins start to denature. So levels of RUBP, GP, GALP, TP, if you're on AQA, OCR, they all start to fall, okay? And if they fall, your net overall product of glucose is going to fall. For example, CO2 concentration. If the CO2 concentration is low, you won't get a lot of conversion of RUBP to GP because there's no CO2 to combine with RUBP to make GP. So GP concentrations start to fall, but you'll get a rise of things. So levels of RUBP will rise, but levels of GP and GALP or TP will fall, all to do with different stages of the Calvin cycle. And that's where the diagrams really help you to understand that. And similarly, light intensity. So this is mainly applicable to light dependent stage reactions. So if you don't get a lot of light dependent stage reactions, you won't get the product, which is your reduced NADP or ATP. Then you won't get any conversion of GP to GALP or TP, or that means you won't get enough glucose at the end. So they're all really well interlinked together. And the best way to remember these things are to constantly draw out these diagrams in your own time, and eventually I guarantee you it will stick in your memory. And the same thing will the same method we'll use in respiration and learning about the 4 main reactions in respiration. If you're an OCR, again, this is an OCR required practical, but on other examples, it can be an application style question. It's basically investigating how pondweed is put in a test tube with sodium hydrogen carbonate as a source of CO2. And you're basically going to alter different of limiting factors, for example, light source. So if you move it closer, you're going to increase the light intensity. And then you would see more bubbles being released, those O2 bubbles, because we saw O2 is used as a product in photolysis. You then collect the O2, and thus you're actually going to be indirectly measuring the rate of photosynthesis. So you can alter the concentration of sodium hydrogen carbonate to increase, decrease CO2 concentrations. You can move the light closer or more further away to decrease or increase light intensity. You can increase the temperature using a radiator or a water bath. So this Pondweed Practical basically sums up all the concepts we've learned. And it's required, again, it's a required practical in OCR, but a really good application style question in AQA and Edexcel. And if you guys want me to, we can go over some of these application style questions. We don't have time for a lot of them, but we can probably do 1 or 2. That's it. Congratulations, you've all finished photosynthesis in 2 hours. I've seen, in my school, I spent almost 2 months learning photosynthesis and I never really actually understood it. So I really hope you guys have understood photosynthesis in 2 hours alone. Respiration, another 2 hours. That's a whole module done already before you've even entered year 13. So congratulations to you. So quick things I just want to mention at the end. The free online biology course will always remain free. That's it. It's always going to be free every week for the first 100 people that can enter, because Zoom doesn't let me allow more than 100 people. So the Zoom link will be on my Instagram. The meeting ID pass is always the same. But yeah, follow my Instagram to get some more updates. I've been really blown away by the response. You guys have been DMing me, commenting, sending me nice feedback. So I really appreciate that. So definitely do give me a follow on Instagram. But there's a problem. We've only got 1 year left if you're in year 12. So we won't be able to cover all the topics. And especially, you've already seen today, the differences in the 3 exam boards mean that I can't cover everything. And we also don't have a lot of time to do any questions and answers. And actually Q&As, questions and answers that from the exam boards are the key to success in A-Level, in my opinion. So we won't be able to get a lot of exam practice at the end. We're only going to be covering common topics like kidneys, the nervous system, things like that. So the next topic, I want you guys to tell me what you want to do. So if you get enough votes, we can do a topic. My thoughts are we'll do immunology. It's been a really highly requested topic. So if you want that to happen, put it in the chat, DM me or something, and I'll be able to start that next time. So we're going to leave out quite a few topics that I can't really cover due to time constraints. But I offer a mentorship scheme, and I've really been blown away. I've almost sold out of all my places on the mentorship scheme this year. So it's basically, you're going to get 1 hour of private tuition of biology, one lesson per week, which is normally worth £20. But I'm going to give you 1 hour of private tuition every week. I'm going to give you exclusive teacher-only resources and past paper questions to help you along. We're going to go through those questions during our 1 hour lesson. I give you useful PowerPoint, any notes I've made. I'm also giving a quarterly mock exam. You also get my contact details, so you can contact me anytime during the week. If you have any questions regarding medicine, your university application, any questions you want to send me, I'm more than happy to answer them. And also, one edition of your personal statement will be hand-marked and critiqued by me before your UCAS deadline. And again, that's a separate service I offer. I'm putting it inside of my mentorship scheme. You can get all of that for only £15 a week. So if you're interested, I've almost sold out. Please DM me soon. I can get you on this mentorship scheme and we can learn biology even better. You can get the grades that you need to get into uni. And that's all we want, really. So that's wrong. I haven't got 6 places left. I've only got 2 left. So if you want to enter the mentorship scheme, you definitely have to inquire now. Personal statement services are on there if you need to look at that. And that's basically it. So tell me how this lesson was. Give me any feedback you have via my Instagram, my email. Basically, all the resources from this lesson and your revision guides and your revision material will all be on the Google Drive link to my Instagram bio. There's no need to sign up to this link. This is from last lesson. Everything will now be on Instagram, so forget about that. Just that's everything you need to know will be on my Instagram. If you need to email me by any chance, please do email me. So guys, we've got a couple minutes left. We've got about 10 minutes left, 5 or 10 minutes. Do you guys have any questions? This can be regarding this topic in particular or any generic questions. Go ahead, this is your time. But I just want to say well done. You guys, loads of you have joined, loads of you have had watched the lessons put on YouTube. You guys have really interacted well with the course. And most of all, you've actually learned photosynthesis, probably one of the hardest topics in A-level in under 2 hours. So I'm really happy for you. The best thing to do now is use the resources I give you on the Google Drive, those diagrams especially. Go through them every day for a couple of days. And by 10 days, if you draw all these reactions, these diagram reactions, once a day, I guarantee you by the end of these 10 days, you'll understand and remember these permanently. So again, these diagrams, they are only a visual prompt. They don't hold any value in the exam to write them as an answer. They prompt your memory and allow you to write down the information from the diagram in words. And that's where you get your marks from. But again, these are the exact same diagrams I used when I did my A-levels, and I find them extremely useful. So the same diagrams we'll be doing for respiration as well. So any questions, guys? This whole PowerPoint will be recorded, so if you didn't understand anything, you can always go back through it at any point. So I will stick around for a couple minutes. If you guys have any questions, I'll be reading the chat and also the Q&A section, so please feel free to add some questions. If you've got any feedback, put it in the chat as well. DM me, email me, give me some feedback. I want to know if this is working for you. And I'll be sticking around for a couple of minutes. If you guys have any questions, please go ahead. If not, thank you very much for joining, and I'll see you guys next week on Thursday at 5 p.m. Again, my plan is to start respiration or immunology, but again, it's up to you guys. Whatever gets the most votes or whatever people say the most, I will go over. But my plans are to do either respiration, immunology. So definitely let me know what you guys want to do next week. But again, thank you very much for joining. I'll stick around for a couple of minutes and I'll see you guys next Thursday at 5 p.m. Thank you. So I've got a question from one of you saying, is this content for CCEA, which is your, I think it's the Northern Ireland exam board? I'm not too sure. So DM me, I'll quickly, after this webinar, have a look at your CCEA specification and I will let you know. I'm pretty sure if you do A-level biology, this is definitely probably in there as well. But definitely send me a DM and I can chat to you about it.